It's a huge pleasure to introduce the second speaker of the morning session today. This will be Gregor Anderluh from Ljubljana, Slovenia. He's the director of the National Institute of Chemistry, but his group is more focusing on membranes, on lipid membranes, on unusual lipids, and they try to find out more about the function and especially the interaction of these lipids with proteins or pore-forming toxins. We will now introduce a really nice talk, pore-forming toxins from molecular mechanisms to nanosensing. Thank you, Gregor.
Thank you very much, Konrad. Hello, everybody. So I'm coming from the National Institute of Chemistry, as it was mentioned. Our group is studying structure function, relationship in proteins in general, and in particular for proteins that damage lipid bilayers, pore-forming toxins. We employ different structure biology approaches, different biophysical approaches. We are able to determine structures of membrane complexes. We are able to generate different model lipid systems. When I was preparing for this presentation, I realized that we use quite a lot of non-ion devices, actually 3 different so far. I'm really happy to be here to present some of our work. Since we work with pores, so these are holes in membranes basically, and we work with unusual lipids and molecular systems, as Konrad highlighted. The talk will deviate from what was heard so far on channels and transporters. And I will, in the first part, show how we can utilize planar bilayer approach and current measurements to study these molecules. And then in the second part, I will show a particular case that is quite interesting in terms of how pores are formed in the membrane. So pore-forming proteins or pore-forming toxins are very efficient in the fact that they can form pores in lipid membranes. This is usually a multi-step process which involves binding to lipid bilayer, oligomerization at the plane of the membrane, and then transfer of part of polypeptide chain across lipid bilayer. I'm showing here 2 nice classical examples, alpha hemolysin pore, which is a beta barrel pore, and hemolysin from Escherichia coli, which is alpha helical pore-forming proteins. So we divide them in these 2 broad groups based on structural element that transfer the lipid bilayer. We study properties of these molecules, so molecular mechanism of how this pore is formed. People study quite a lot effect on cells. This is not clear yet why cells are killed due to action of these toxins. This is the final outcome sometimes. And they can be used as molecular tools due to their unique properties. This is the gallery of some of pore-forming toxins of model systems that were studied over the years. As mentioned, alpha helical or beta barrel ones. We need structures. We need structure of soluble monomer in order to understand how these final assemblies form. From this gallery, you can see that they're quite diverse in structure. They're quite diverse in size of this final oligomeric assembly that is formed. Some of them are small, couple of nanometers in diameter, but some of them are quite large, so several tens of nanometers in diameter, forming really huge pores. One feature that is common to all of them is also that quite often they are multi-domain proteins. So each domain has a different role in pore-forming mechanism. For example, this is cholesterol-dependent cytolysine family, where one domain, domain 4 at the bottom of the protein, is used for attachment to lipid bilayer, and other domains then participate in protomer-protomer contacts and formation of this huge beta barrel that transfers the membrane. As mentioned, because of their properties, they can be exploited as molecular tools in cell biology, for example, for selective permeabilization of cells, or for labeling of different lipids. If you just use the domain, which is a non-toxic protein, then you can label different lipids, cholesterol, sphingomyelin, and so forth. And of course, they can be used as sensors in sensing technologies. The nicest example is Oxford Nanopore Technologies DNA sequencing. But other modalities of sensing were also described in the literature. Conformational changes in enzymatic analysis, for example, can be captured by measuring currents through such pores, or discrimination of peptides from a mixture of different peptides. So they're quite used in sensing approaches, and one of the aims of our group is also to discover novel types of pores that could be used for sensing approaches. So we used a lot planar lipid bilayer approach, where a membrane is formed between the two compartments, and then the ion currents can be measured. So if you add to such a system a pore-forming protein, then depending on the family, different outcomes may be realized. So this step-like increases implicate formation and insertion of a pore. And in some cases, you can see that this is quite a homogeneous population of pores with narrow distribution of conductances. But in some cases, this is quite diverse. There's some imagination even need to be used in order to analyze properly this data. And in terms of noise, they can be quite different. This is one of the fewest approaches actually that we can use to study directly activity of these molecules. That's why we value it quite a lot, and quite often we can resolve from this kind of traces properties of these pores before we have any structural information. I will show a couple of examples. For example, these kind of pores are formed by beta-barrel proteins such as lysine here, which form pore by 9 molecules and form this beautiful beta-barrel narrow tube across lipid bilayer. Consequently, so it's a quite rigid structure. Consequently, the traces are do not exhibit a lot of noise, and they're quite homogeneous. On the other hand, this is family of actinoporins, pore-forming toxins from sea anemones, that they form pore by alpha helices, by cluster of alpha helices. And the result, this kind of traces are a result, first of all, of structural heterogeneity, because stoichiometry of these pores can vary from 7, 8, 9 MERS. And also, it's quite interesting observation. This is our recent work on when we determined the structure of one of these pores. So this is octameric complex that was extracted from the membrane by detergents. And you can see that apart from protein, there's also quite well-defined lipids at different positions within the protein. And some of the lipids actually fill the area between the protein molecules, and they're integral part of oligomeric complex. And there was one lipid that protrudes between the helices into the lumen of the channel. So it's highly dynamic complex, and that's why also the traces that we observe with these pores are quite non-homogeneous. We analyzed this pore in a greater detail, in particular at different lipid compositions. When we included cholesterol in these membranes, the extracted membrane part was really nicely visible, and each protomer covered 15 lipids. So each protomer sits on 15 lipids, and we could resolve these lipids very nicely. We could assign different roles to these lipids. So some of them are structural, which allow initial binding of protein to the membrane. Some of them are bridging, just at the edge of this complex, bridging through protomer, lipid protomer interaction, the whole complex. And we could also observe cholesterol nanodomain, which was hidden below the cap region next to the alpha helical region. So it's really a beautiful structure that shows how protein is embedded in lipid bilayer, and how lipid membrane is organized around the alpha helical region that form the conductive pathway. Another example is a bit larger protein, perforin. Perforin is a key protein in the immune system. It forms pores in cancer cells or viral infected cells and killing them. It is a protein that is dependent on calcium ions, so nothing much happens. And when you add calcium ions, then pores will begin to form. So also for this study, we didn't have much structural information. We observed that there was a range of sizes of these pores with different noise, and depending also of the lipids that we used for membrane preparation. So quite heterogeneous picture. And it was thought that perforin only forms this kind of pores. So large pores where more than 20 molecules come together to form this larger than 10 nanometer pore in the diameter. So our idea at the time was that this different situation with regards to pore formation depends on the nature of lipid membranes. So in more fluid membranes, these oligomers that are not yet full rings can already insert, while in more rigid membranes, you need the whole ring to then collectively penetrate the membrane and form this final pore. And exactly this situation was later then observed with AFM experiments when they observed all of these different intermediates. But in recent years, we study a lot smaller proteins, NEP1-like proteins, and actinoporins, as mentioned, as shown. So they both are belonging to a bigger family of proteins with similar topology, a beta sandwich topology with loops at the bottom, and different binding sites for lipid head groups or for sugars of the cell surface. For actinoporins, we quite understand this formation of membrane damage. So they are attached to membranes, as mentioned before, via sphingomyelin, and they then transfer this N-terminal region across the membrane to form this alpha helical pore that I showed just before. So NEP1-like proteins are a bit different. So the beta sandwich architecture is conserved, but these other parts and loops at the bottom, but the other parts are not so well conserved. One interesting feature of these molecules is that they would only work in plant sphingolipids. And no other membranes will be a good substrate for binding and membrane damage. Plant sphingolipids are different from animal ones. They have a ceramide part, phosphate group, and then inositol, and then range of sugar molecules that start with glucuronic acid and some hexoses. So the number of hexoses can vary between different plants. So we have series A, B, C, D, and so on. They can be branched and so on. But you need this kind of lipids in order to see binding and activity of these molecules. So they are produced by microbes that infest plants. We can prepare them in Escherichia coli. We can inject them in plant leaves, and as you can see here, then necrosis formed in a couple of days, so completely damaging plant tissue and killing the cells. You cannot obtain these lipids anywhere else, so we purify them. This is our lipid extract. And these GIPCs, they're called glycosyl-inositol phosphoceramides. These GIPCs are in this part of the TLC plate. So if you don't have this lipid, this family of proteins will not work. So it's quite challenging to understand how these molecules damage lipid membranes. But after we were able to extract these lipids from leaves of plants, we were able to generate giant vesicles. We were able to generate some model lipid systems, and we could study the activity. This is the result of Orbit 16, just recent results with these proteins. So we can generate a lipid bilayer that is quite stable for a long time. And upon addition of the protein, then after some time, we can observe this kind of traces. So in contrast to beta barrel proteins such as lysine, which form step-like increases in current, this is just something, a lot of noise and transient pore openings. And always after some time, membrane will collapse after several minutes of recording. We have two different lipid preparations from two different plants, from tobacco or from leek. And in both cases, both GIPCs show similar behavior. So a lot of noise and not very nicely resolved increases in current as observed for beta barrel proteins. So how do you analyze this kind of traces? It's really terrible. So one parameter that we thought was the time that passes from addition of protein to first changes in current. And we plot this here. And you can see that these two different membranes differ a little bit in this parameter, but not much. We also, so this protein family, NEP1-like proteins are a big protein family. There's almost 2,000 sequences known. This is another one which forms from another organism which forms similar traces. So a lot of noise, transient pore openings, and always collapse of the membrane at the end of the measurements. We supplement this kind of traces with other approaches. So with giant vesicles, we were able to show that these pores that are formed are small. These are different sized dextrans, fluorescently labeled. When added to exterior vesicles, and you can see that smaller ones can equilibrate across the membrane in the presence of protein, but larger ones do not. And this is interesting observation also. Quite often we get multilamellar vesicles, giant vesicles, and in this system always only the outmost lamella was permeabilized, but not the inner ones, indicating that protein could not enter through self-made pores. And protein is 4 nanometers, 4 to 5 nanometers in diameter. So also confirming that pore size is small. And finally, we also image these pores with atomic force microscopy. So this kind of lipids, they form phase-separated domains. We saw binding of protein, these bright dots in liquid disordered phase first, and accumulation at the edges of the domain, and then the oligomeric assembly is formed within the liquid ordered domains. It was a very dynamic system, as you can see here from consecutive scans of the same area where some of the oligomeric assemblies were removed by the scanning and some new formed, indicating that protein is not inserted deeply in the membrane. The oligomers were quite diverse in size range from 6 to 12 nanometers in diameter, and protein was just sitting on the membrane. So this height of the protein was around 3 nanometers, which we confirmed also by other approaches. So this family of proteins, collectively, what we know so far is, so they're similar to actinoporins, which interact through sphingomyelin to form the final pore. But these molecules associate with plant sphingolipids. They oligomerize at the plane of the membrane, do not penetrate deeply, and this final pore, we don't know yet how it looks, but we believe that maybe some transient openings are formed due to clustering of these protein molecules and significant interaction with head groups of glycosylinositol phosphoceramides. So in the future, we wish to study properties of these membranes, and also we'd like to get more insight into the final oligomeric assembly by structural approaches. I would like to thank again for the invitation and for your attention. I need to thank my colleagues from the department, three excellent PhD students, Maria, Gasper, and Tina, who performed experiments that were shown here. Marietka is structural biologist in the group, and Franci helped us with molecular modeling, which I didn't show here. And we have some collaboration also with Oxford Nanopore Technologies. Thank you very much.
Yeah, thank you so much, Gregor, for this really nice talk. Something a bit different now. There's the first question.
Thank you. That was very interesting. I'm wondering about this result with the GUVs, where you showed that actually it cannot penetrate. That would mean that the outer membrane. So that would mean that this membrane stays intact.
Yeah.
In contrast to the membrane on the orbit 16, which invariably ruptured.
Yeah.
So this suggests the question: what is the role of the membrane potential for the actual membrane rupture? Have you looked at the voltage dependence of the kinetics of membrane rupturing?
No, not yet. That's a good point. Actually, what we know is that it's initial interaction of protein with membranes is electrostatic in nature, definitely. So if you use high salt, then they will not bind, they will not damage membranes.
So it means they have a surface charge, these things.
Yeah.
And I mean, it could be that the pore-forming mechanism is some sort of reordering of the lipids that makes them more susceptible to an electrical field to break down. So in fact, you have some sort of increased propensity for electroporation in a certain way.
Okay, it could be. So our idea is that proteins, when they are attached to membrane, we know from modeling they form multiple contacts with different GIPCs. So it's not one-to-one, but they're quite engaged with the protein, which is highly positively charged, and these lipids are highly negatively charged. So it could be that this reorganization of lipids then also help in.
So it reminds me of something we did with some nanodots at some point that also had a strong charge, and Tuljan may remember these these recordings, which were never published, but it's an interesting idea. Yeah. Thank you.
Okay, thank you for the comment.
Thank you so much. Another question. All right.
We learned yesterday from Alexandro, for example, from Urea Biosciences, that the membrane thickness of organelles is less thick than the normal cell membranes. And I'm wondering if the membrane thickness is also a parameter that you guys adapt specifically. Is this a parameter that you consider at all?
Not yet. It's a good comment again. We could think about that. So the head groups are quite bulky, and but in modeling, you know, they adopt different kind of conformations at the membrane. So membrane is not much thicker than if you use like PC or sphingomyelin. But with regards with acyl chains, this can be quite a factor as well, because these plant sphingolipids, they have very long chain acyl chains, like 24 carbon atoms, and they interdigitate between the monolayers. So it could be that this kind of features also affect the activity. I mean, we use exactly the same membrane for beta barrel proteins insertion. I showed this trace for lysine, and it didn't bother this protein, so it inserted quite efficiently into this membrane.